Good afternoon. Today's lecture topic is the structure and functions of biological membranes. We will discuss the following issues: the main types of biological membranes and their functions, the chemical composition and structure, basic properties of biomembranes, mechanisms of transport of substances through biological membranes. Learn the definition. Biological membranes are complex supramolecular structures that surround all living cells and form closed special compartments within them, called organelles. The main types of biological membranes are shown in figure. The membrane surrounding the cytoplasm of the cell from the outside is called cytoplasmic or plasma membrane. It is indicated in the figure by the number 1. Name of intracellular membranes are usually derived from the name of subcellular structures restricted or formed by these membranes. There are nuclear, it is indicated by the number 2, mitochondrial, number 3, lysosomal, number 4, Golgi apparatus, number 5, endoplasmic reticulum, number 6, and other membranes. Familiarize yourself with some examples of functions of membranes and memorize them. Plasma membrane limits the contents of the cell from the external environment, provides contact with other cells, receiving, processing, and transmission of information inside the cell, maintaining a constant internal environment. The nuclear membranes, internal and external, form the nuclear envelope, which separates the chromosomal material from the cytoplasmic organelles. Through the pores of nuclear envelope, the proteins and nucleic acids are transported into the nucleus and from the nucleus. Mitochondrial membranes, internal and external, are responsible for transforming energy in oxidative phosphorylation ATP synthesis. Lysosomal membranes protect the cytoplasm of the cell from the hydrolytic enzymes, prevent self-digestion, autolysis, of the cells, contribute to the maintenance of a constant pH in lysosomes. The membranes of the endoplasmic reticulum are involved in the formation of new membranes, carry out the synthesis of proteins, lipids, polysaccharides, oxidation of hydrophobic metabolites, and xenobiotics. Note: all membranes have some common properties that are characteristic of their organization and composition. These properties include being composed of lipids, proteins, and carbohydrates, having a flat closed structure, asymmetry, having both an internal and external surface, selective permeability. The basic component of any membrane is the lipid bilayer, which is a double layer of lipid molecules. Since the basis of biomembranes is formed by a lipid bilayer, it is necessary to focus more on the properties and classification of lipids. Lipids are a large group of compounds that significantly differ in their chemical structure and biological function. Common characteristics of lipids include insolubility in water, good solubility in non-polar solvents such as ether, chloroform, and benzene, the presence of hydrocarbon radicals in their structure, prevalence in living organisms. Remember that most of the lipids in the membranes of mammals are phospholipids, glycolipids, and cholesterol. All of them are derivatives of higher fatty acids and monatomic or polyatomic alcohols. The fatty acids that make up the lipids of biomembranes can be divided into 3 main groups. 1. Saturated fatty acids with no double bonds in their hydrocarbon chains. The most common examples of these acids are palmitic acid and stearic acid. Their empirical formulas can be seen on the slide. These saturated fatty acids are produced in the human body. 2. Monounsaturated fatty acids have single double bond in their hydrocarbon chains. Oleic acid and palmitoleic acid are examples of this type of acid. These fatty acids can also be made in the human body. 3. Polyunsaturated fatty acids which have 2 or more double bonds in their hydrocarbon chains. Examples of such acids are linoleic acid with 2 double bonds, linolenic acid 3 double bonds, and arachidonic acid 4 double bonds. These acids are not produced by the human body and must be obtained from food. Linoleic and arachidonic acids belong to omega-6 fatty acids. Linolenic acid is an omega-3 fatty acid. The letter omega indicates the carbon atom farthest from the carboxyl group. The number indicates the position of the double bond nearest to the omega carbon atom. Pay attention to the main structural features of fatty acids, which make up natural lipids. They contain an even number of carbon atoms, between 16 and 20. The double bond in unsaturated fatty acids is located between the 9th and 10th carbon atom. Double bonds in polyunsaturated fatty acids are separated by methylene groups. That is, they are non-conjugated bonds. These double bonds are in a cis configuration, which causes the hydrocarbon chain to bend. Phospholipids in the membranes are divided into 2 groups: glycerophospholipids and sphingomyelins. Glycerophospholipids are esters of glycerol with 2 fatty acid and phosphorylated amino alcohol. Sphingomyelins are derivatives of ceramide, the amide of sphingosine and a fatty acid, and a phosphorylated amino alcohol. Glycolipids are also derivatives of ceramide, containing 1 or more residues of monosaccharides. These lipids are divided into cerebrosides, containing a single monosaccharide residue, and gangliosides, containing chains of several monosaccharides, one of which must be sialic acid, which has a negative charge. The composition of glycerophospholipids includes phosphatidic acid, which is an ester of glycerol and 2 fatty acids, and phosphoric acid. These lipids also contain one of the amino alcohols, most often choline, ethanolamine, or serine, which attaches its hydroxyl group to the phosphoric acid residue in phosphatidic acid. The most common glycerophospholipids in membranes are phosphatidylcholines. The formula of one representative is presented on the slide. The fatty acid residues can be different, but the second hydroxyl group of glycerol must be linked with the residue of an unsaturated fatty acid, in this case linoleic acid. The composition of a sphingolipid molecule includes a ceramide residue, an amide of sphingosine, and an unsaturated fatty acid. The first hydroxyl group of this residue can be bonded to phosphocholine via a phosphate group in sphingomyelin, a monosaccharide, for example glucose or galactose, in cerebroside, or an oligosaccharide in ganglioside. The slide shows an example of sphingomyelin containing an oleic acid residue. An example of a cerebroside is galactoceramide, which contains a galactose residue in the first position and a linolenic acid residue in its ceramide composition. Other galactocerebrosides can contain residues of other unsaturated acids. Along with phospho and glycolipids, the bilayer of biological membranes may include cholesterol, a monatomic cyclic alcohol. It is one of the main components of the plasma membrane of animal and human cells, and may also be present in smaller quantities in mitochondria, Golgi apparatus, and nuclear membranes. There is a lot of cholesterol, especially in nervous tissue. There is no cholesterol in plant cells. Cholesterol, due to its hydroxyl group, is able to form esters with fatty acids. Cholesterol esters are not included in biomembranes. The characteristic feature of membrane lipids is their amphiphilicity, presence in the molecule of both hydrophobic and hydrophilic sites simultaneously. The hydrophobic part of the molecule is represented by the hydrocarbon chain of fatty acids and the side chain of sphingosine. The hydrophilic regions are represented in phospholipids by phosphorylated amino alcohols and in glycolipids by sugar residues. The amphiphilic nature of lipids determines their behavior in the aquatic environment. Adhesion of hydrophobic regions of molecules leads to formation of ordered closed structures, micelles. In these micelles, the hydrophobic parts are protected from water and the hydrophilic parts are immersed in an aqueous medium. The amphiphilicity of cholesterol is weak. Its cyclic structure and side chains are hydrophobic, but only the hydroxyl group is hydrophilic. Therefore, cholesterol molecules are embedded in the empty spaces between the hydrocarbon chains of fatty acids in the hydrophobic part of the bilayer, and the OH group is adjacent to the hydrophilic heads of phospholipids. This structure, stabilized by non-covalent hydrophobic interactions between nonpolar groups, is thermodynamically very stable and underlies the formation of biological membranes. Membrane lipids act as a solvent for membrane proteins, creating a liquid medium in which they function. According to their functions, proteins are divided into 4 categories. 1. Structural. 2. Catalytic. 3. Receptor. 4. Transport. The number of proteins in the membranes depends on the type of the membrane and its function, and can vary significantly. For example, in myelin membranes designed to isolate nerve fibers, proteins make up only 25% of the membrane mass. In mitochondrial membranes associated with oxidative phosphorylation processes, proteins account for approximately 75% of their mass. The proportions of proteins and lipids in the plasma membrane are approximately equal. According to the degree of influence on the structure of the bilayer and the strength of interaction with it, membrane proteins are divided into integral and peripheral. Integral proteins are deeply embedded in the membrane structure and cannot be removed without destroying the membrane. They are fixed in the bilayer due to hydrophobic interactions with the hydrocarbon chains of fatty acids. Peripheral proteins are located on the surface of the bilayer and can be extracted with salt solutions or water. They are globular, hydrophilic structures that are fixed to the surface of the bilayer by ionic interactions with polar parts of phospholipids and integral proteins. Carbohydrates in membranes are not separate substances, but are found only as part of proteins, glycoproteins, or lipids, glycolipids. The length of carbohydrate chains ranges from 2 to 18 monosaccharide residues. Most of the carbohydrates are located on the outer surface of the plasma membrane. The functions of carbohydrates in biomembranes include the control of intercellular interactions, maintenance of immune status, reception, and providing stability to protein molecules in the membrane. The figure shows a diagram of the structure of a plasma membrane fragment. It is based on a double layer of phospholipids and glycolipids. The hydrophobic parts interact with each other, while the hydrophilic parts come into contact with extracellular or intracellular aqueous media. Cholesterol molecules fill the empty spaces formed as a result of bends in the carbon chains of polyunsaturated fatty acids, phospho, and glycolipids. Integral proteins penetrate the lipid bilayer. Peripheral proteins are linked to the outer or inner surface of the bilayer. Carbohydrate components are found in glycolipids and glycoproteins on the outer layer of the membrane. The closed lipid bilayer determines the main properties of membranes. Among them, fluidity is important, which depends on the ratio of saturated and unsaturated fatty acids in the composition of membrane lipids. The predominance of saturated fatty acids reduces the fluidity of the membrane. The predominance of unsaturated fatty acids gives the membrane greater fluidity. The fluidity of membranes is also influenced by cholesterol content. Cholesterol embedded between fatty acids seals them and increases membrane rigidity. The figure schematically shows the structure of a bilayer, with a predominance of saturated and unsaturated fatty acids in its lipid composition. Hydrophobic chains of saturated fatty acids are oriented parallel to each other and form a rigid crystalline structure. Unsaturated fatty acids with a curved hydrocarbon chain break the compact packing and increase fluidity of the membrane. The presence of free space between lipid and protein molecules in a biomembrane determines its next property. Lateral diffusion, the free movement of molecules relative to each other in the plane of membranes. Unlike lateral diffusion, the ability of molecules in biomembranes to traverse diffusion, transition from the outer layer to the inner layer and vice versa, is limited. This contributes to maintaining asymmetry, structural and functional differences between the outer and inner layers of the membrane. Examples of membrane asymmetry. In the plasma membrane of an erythrocyte, phosphatidylcholine and sphingomyelin predominate in the outer layer, while phosphatidylethanamine and phosphatidylserine predominate on the inner layer. The carbohydrate parts of glycolipids and glycoproteins are found on the outer surface, forming a continuous coating, glycocalyx, on the cell. But there are no carbohydrates present on the inner side of the membrane. The main properties of biomembranes also include the impermeability of a closed lipid bilayer to most water-soluble molecules and ions. This is due to the presence of a hydrophobic phase in the bilayer. As already mentioned, one of the main functions of membranes is their participation in the transfer of substances. This process is provided by three main mechanisms: simple diffusion, facilitated diffusion, and active transport, which are shown schematically in the figure. Membrane transport of substances also differs in the direction of their movement and the amount of substances carried by this carrier. 1. Uniport, transport of one substance in one direction depending on the gradient. 2. Symport, transport of two substances in the same direction by a single carrier. 3. Antiport, movement of two substances through a single carrier in opposite directions. Let's consider the main features of membrane transport mechanisms. Simple diffusion is the transport of substances through a membrane without the involvement of specific mechanisms. Transport occurs down a concentration gradient without input of energy. Examples of substances transported by simple diffusion include small molecules such as water, carbon dioxide, oxygen, urea, and hydrophobic substances with low molecular weight. The rate of simple diffusion is proportional to the concentration gradient. Facilitated diffusion is the transport of substances across membranes by protein channels or specific carrier proteins. The transport occurs down a concentration gradient without input of energy. Monosaccharides, amino acids, nucleotides, glycerol, and some ions are transported. Unlike simple diffusion, facilitated diffusion has saturation kinetics. The kinetics of saturation is as follows. At a certain saturating concentration of the transferred substance, all carrier molecules participate in the transfer, and the transport rate reaches a limiting value. The graph of the dependence of the transfer rate on the concentration of the substance has a hyperbolic shape. This resembles the dependence characteristic of enzymes. With an increase in the concentration of the substrate, the rate of catalyzed reactions gradually increases to a certain value, after which it becomes constant. Active transport also requires the involvement of specific carrier proteins. However, the transfer occurs against a concentration gradient and therefore requires energy. Sodium, potassium, calcium, and magnesium ions are transported by active transport across the cell membrane. Protons are also transported across the mitochondrial membrane by active transport. Saturation kinetics is characteristic of active transport of substances. Depending on the energy source, there are two types of active transport. 1. Primary active transport, in which the transfer of substances is associated with an energy-generating reaction. As a rule, such a reaction is hydrolysis of ATP. 2. Secondary active transport, in which the transfer of one substance against a concentration gradient is associated with the passive transfer of another substance along its concentration gradient. An example is the mechanism of glucose reabsorption in the renal tubules. Glucose penetrates into the epithelial cell against the concentration gradient, along with sodium ions which move along the concentration gradient. An example of the transport system that carries out active transport of ions is sodium, potassium adenosine triphosphatase, sodium potassium ATPase, or sodium potassium pump. This protein is located within the plasma membrane and catalyzes the hydrolysis of ATP. The energy released by the hydrolysis of one ATP molecule is used to transport 3 sodium ions from the cell into the extracellular space and 2 potassium ions in the opposite direction. This results in an ion concentration gradient between the cytosol of the cell and the extracellular fluid. Because ion transport is not symmetrical, an electrical gradient also appears. Therefore, the generated electrochemical potential is the sum of these two factors. Not only low molecular weight substances can pass through biomembranes, but also high molecular weight compounds and even relatively small particles. The transfer of particles through the plasma membrane occurs as part of a membrane vesicle and is called vesicular transport. In vesicular transport, endocytosis and exocytosis processes are distinguished depending on the direction of transport and the nature of the transported substances. Endocytosis is the process of transferring particles into a cell. There are several types of endocytosis: pinocytosis, the capture and absorption of water-soluble macromolecular compounds by a cell; phagocytosis, the capturing and absorbing of solid particles by a cell; receptor-mediated endocytosis, when capture is preceded by binding to specific receptors on the plasma membrane. This type of endocytosis is often used in immune processes. In all these cases, the plasma membrane initially embeds itself in the cytoplasm at the site of substrate penetration. Then, it gradually deepens until it becomes a membrane vesicle containing the substrate and is completely located within the cytoplasm. Exocytosis is the transfer of particles and large molecules from a cell. The most common type of exocytosis is secretion. This is the elimination of soluble compounds from the cell, which is one of the functions of this cell. Accumulation of substances in the cell takes place in the form of secretory vesicles. The membrane of the vesicles then merges with the plasma membrane, so that the contents of the vesicle are outside the cell. High and low molecular weight compounds, such as enzymes, hormones, and mediators, can be secreted this way. If solid particles are removed from a cell, this type of exocytosis is called excretion. An example of excretion is the removal of reticular substances, aggregated organelle residues, from reticulocytes at the end of erythropoiesis.